Thank you. So it's my pleasure to welcome Dr. Chiara Herzog. She's a PhD and postdoctoral researcher at the European Translational Oncology Prevention and Screening Institute at the University of Innsbruck, Austria, and an honorary research fellow in University College London, UK. Her background is in molecular medicine, and she's kindly agreed to present today on her research looking at the potential of routine cervical samples for detection and risk prediction of breast and ovarian cancers. And I'd like to thank Dr. Herzog for agreeing to present today. Thank you.
Great, now I had to find the unmute button. Thank you very much, Alison and Christian, for inviting me and giving me the opportunity to present my work, especially over the last 2 years. The scientific exchange has been a bit reduced, obviously, because all sorts of conferences have been massively scaled back. So I'm really pleased to present today. So I'm going to give a kind of brief introduction into epigenetics and why we are interested in using epigenetic footprints for cancer detection and risk prediction. Just a very brief overview of my talk. I'm first going to introduce epigenetics as a concept and what it consists of, and I'm also going to go into a bit more technical aspects, so the kind of tools we have available for analyzing epigenetics. I'm also going to briefly introduce our research question at UTOPS. So Alison has already explained the abbreviation. It's the European Translational Oncology Prevention and Screening Institute. It's quite a long name, so we abbreviate it to UTOPS. And of course, I'm going to talk about the WID tests, which are our tests for detection of ovarian and breast cancer in cervical samples. And I'm going to give a brief outlook of the future research question we will address. So first of all, what is epigenetics? So I think I have a bit of a quite diverse audience here today, so I thought I'd start by introducing it a little bit. Basically, epigenetics consists of all the modifications and spatial arrangements of the DNA at different levels. So obviously we have a certain sequence, but the DNA is not naked, so to say, in our cells. It's neatly packaged and modified. So at the kind of quite small molecular scale, we have DNA methylation, which consists of these additions of methyls at cytosines in the CG dinucleotide context. So every time you have a CG context, the cytosine before the G can be either methylated or not. Then the DNA can be further wrapped around histones. So these are kind of structural proteins that help to organize the DNA. These can also then be modified. So you can either have methylation or acetylation. And depending on these markers, the histones can then be more condensed or more open. And altogether, that then forms the chromatin structure that you can observe in the nucleus. These epigenetic features are really important, actually, because they define cell state transitions and phenotypes, and they're what allows us to have the same shared DNA in all of our cells with very different functions. So if you imagine a hair follicle cell will have a very different function from a breast cell. So yeah, epigenetics is kind of a master regulator of these phenotypes. A majority of these studies has actually previously been done during development, which has enabled us to discover the role of DNA methylation in particular. So shortly after fertilization, actually the majority of CpGs lose methylation. So you can see here kind of a schematic overview of the percentage of CpGs that are methylated in a cell. And this reduces drastically. So we kind of have a ground state of the cell, which is almost exclusively unmethylated. And then with increasing development, we have actually, again, a rapid methylation of almost the entire genome, except some protected areas that I'm going to talk about in a little bit. But yeah, so basically the entire genome is then methylated, except these high density protected regions. And actually, this is where I'd like to introduce my first poll. So perhaps this is a bit going back to your education, but yeah, so what are the regions with high frequencies of CPGs called? Are they called promoters, shores, islands, or enhancers? And this is, I'm not doing an exam. This is just making it a bit more fun and interactive than just me speaking at the screen for an hour.
I'll leave the poll up for about
That's
10
fine.
seconds.
Yeah, I'll just leave it going on in the background. But yeah, basically, we have this. So throughout the somatic tissues, which is actually mostly what we're going to talk about. So this is anything after development. You have almost exclusive methylation. Okay, so this is a very good. So the question, the answer is actually the islands. However, promoters is pretty much also true, because actually most of the promoters, so gene promoters, will include such a CPG island, and they're really important regulatory elements. So yeah, well done. Exactly. So the CPG methylation in these promoter sites or in the islands is often associated with gene transcription. So if there's very little methylation, then the genes are often active, although it's not always as clear as that, and it's getting a bit more complex as we do more research into this. But yeah, so basically we see these fluctuating patterns of methylation, especially in the germline. We then lose again methylation, but this is not something I'm going to talk about today. So I've already now briefly touched upon the kind of more regulated DNA methylation changes across lifetime. However, DNA methylation is also influenced by exposures, some of which we can control, some of which we can't. So obviously aging is something that affects us all, but it's known to heavily influence DNA methylation patterns. And actually you can generate so-called epigenetic clocks, where you can say really accurately how old the person is that donated a sample. But it's also influenced by more conscious choices that we make. So diet, smoking, or for example, alcohol or drug intake can all modify DNA methylation. Something that we are quite interested in is also hormone exposure. So depending on, for example, the number of pregnancies or lifetime hormone exposure, the DNA methylation patterns can change. Also, environmental factors or the microbiome can change DNA methylation. So really the DNA methylation seems to be a very good marker for basically anything that a human has been exposed to in their lifetime. And this is particularly interesting because that means that DNA methylation actually integrates the genetic and non-genetic risk factors that we are exposed to in a lifetime and records them in a signature. So the genetic component obviously comes from the underlying sequence that we have recorded in our cells. And the non-genetic risk factors come from, for example, lifestyle choices or other things that are perhaps beyond our control. And what is especially important in this context is that aged cells, but also cells at cancer risk, display distinct patterns. So you can actually also tell really well if a sample is a tumor or not from DNA methylation patterns. What we often see is, and I've tried to illustrate this a little bit here. So if we think that this is an island or a promoter in light blue, there's usually less methylation, as I've already mentioned, compared with the rest of the genome here in dark blue, where we have a bit more. However, with aging, but also with increasing cancer risk, we see kind of a hypermethylation, so an increase in these methyl groups, but the genome-wide, we lose methylation. But the important thing from this to take home is that DNA methylation is dynamic and it can be altered by interventions. So you can theoretically erase these aging or cancer signatures, and these might also have a causal effect. One thing where we know this quite well is from reprogramming. So if you take cells from an adult, you can then reprogram them to pluripotent stem cells, which is also based on this alteration of DNA methylation patterns. So I hope this has given you a bit of an insight into why we think DNA methylation and epigenetics is a really interesting tool to look at risk monitoring and detection. So it's a dynamic marker. Unlike single nucleotide polymorphisms or gene mutations, DNA methylation can account for dynamic changes in risk and lifetime exposure. So a single nucleotide polymorphism score, for example, is quite good for predicting risk for a general population, but it cannot tell you about a certain individual and their individual personalized risk. DNA methylation integrates the exposures of a cell and leaves an epigenetic footprint on the DNA that we can then measure. And this is what I'm going to talk about next. And for analytical problems, it's actually relatively stable compared to RNA and proteins. So it's a lot easier to work with. Okay, so this was a brief introduction. I'm now going to talk about the analysis tools that we have available. And actually, this is where I'd like to start my second poll, just to get a bit of an background in the audience. So which type of analysis you're usually doing. So this is, it's just for me to kind of understand what kind of audience we have and what you do. So in terms of the analysis of DNA methylation and epigenetics markers, one of the issues is that we cannot directly read out whether a C is methylated or not. So the majority of tools at the moment are not capable of doing this. There are some more novel approaches, but these are not routine yet. I'm just going to wait for the result. Okay, cool. Okay, that's good to know. So yeah, I'm going to talk about all of these, but it's good that I try to include a bit of a balanced result. Okay, great. So yeah, as I've already mentioned, we can't directly read out these methylated or tagged Cs, so we need to do something to them. So what has been kind of the gold standard for this in the last decades really is bisulfite conversion. So what happens is when you expose a sequence to sodium bisulfate, it's a deaminating agent. Basically what happens is that all Cs are converted to uracils, except those that are protected, which are the methylated Cs. So these cannot be converted. So this kind of distinguishes between methylated and unmethylated Cs. And then when you do a PCR amplification of that, you can basically, so these U's are then converted to T's, and you can then compare this with a reference genome, and okay, here should be a C and it's still a C, so this one was methylated. This one should actually be a C, but it's now a T, so it was likely unmethylated. So this is kind of the underlying principle for all of the techniques I'm going to introduce. So there's a variety, all have their own advantages and limitations. Very commonly used are arrays. So for example, there's the Illumina methylation epic, which covers around 450 to 850,000 CpGs. This might sound like a lot. However, I probably should have mentioned this earlier. There's around 26 million CpGs in the human genome. So you can imagine this is a very small proportion of the total amount of CpGs. They're quite similar to microarrays, and there are specific probes for each single CpG site. I'm going to talk about each of these in a little bit more detail soon. Sequencing is more expensive at the moment, at least, but it covers a lot more CpGs than arrays. So in theory, you can convert the entire genome and then just sequence everything and cover basically all of the CpGs. And what you also have is obviously the information about CpGs next to each other. So unlike for the array where you only look at one specific site, in a read of, for example, 150 base pairs, you might have multiple CpGs, and then you can say, okay, within this read, how many of them are methylated and what's the relationship between them. And then we have Methylite PCR, which is basically a real-time PCR methylation specific. It's very cost effective and offers targeted assessment of regions. So of course we can't do the entire genome, but sometimes all you need is a certain region. And this is also something that we are working on. Keep your eyes peeled for some future interesting publications, but I'm going to keep this one out of the current talk. So just very briefly for the arrays, because these are the tools that we are mainly using for the work that I'm presenting today. If you have an unmethylated locus, so here this is kind of symbolized by a blank tag. So these would be unmethylated, and we know from the previous slide that I've shown that bisulfite conversion ultimately makes this a T. You would then have a probe that can anneal to this region after it's been converted. And you see here, actually there's two sites, but we're only able to look at this one for this specific probe. And a complementary a nucleotide can be extended, and this one is an A, which is has a red fluorophore. If that specific locus actually were methylated, so that's the other scenario, then the C stays a C, and the complementary nucleotide is a G, which has a green fluorophore. So essentially what we're doing on this array is we have 850,000 of these probes, and they will have either a red or a green signal. And this is very efficient for analysis and is also quite standardized. So that's, I think, why it's a very popular technology. The other technology that is on the rise now is also sequencing. So I've already shown this little diagram of bisulfite conversion and amplification. So what you would do in this scenario is not to have a probe anneal, but rather to sequence your fragment. And what you can do then is you have this bisulfite treated sequence from your machine. And you can align it to the reference genome. So you then see, okay, this C stays the same, so it must have been methylated. This one is actually now a T, so it must have been not methylated. But as you see here, you would, for example, have a read with multiple Cs, and then you can see which ones were methylated and which ones were not. So this is what's called phasing. And just very briefly, because I wanted to introduce it as it's a really neat technology, is Methylite PCR. So there's a couple of different strategies, but it's essentially based around the fact that you would amplify a fragment of DNA, which is within your region. So all of this happens after bisulfite conversion. And you can then design primers, for example, that are specific to a methylation site. And then you have an oligo that can hybridize and would emit a fluorescent signal. So the more of this fragment you can amplify, the more fluorescent you get. And then you can quantify essentially the amount of methylation in your region. So yeah, just wanted to introduce these technologies. Great. All right, so now I'm going to talk a bit more about our actual research. We have a very specific vision, which is to make women's cancers a disease of the past by 2040. Of course, this is quite ambitious, but there won't be any progress without ambition. So we focus on 4 gynecological malignancies. So we look at breast, ovarian, cervical, and endometrial cancers, which together account for more than 50% of all women's cancers. Our work is based around 4 pillars, and they're obviously all interlinked, but they're slightly different projects. So obviously we want to understand cancer development and factors that drive cancer development. The work that I'm going to present today is about the prediction. So can we identify those individuals with increased risk and then do something about it? So we actually capture the disease before it starts. Another aspect is prevention. So this is obviously closely linked with the prediction. So if we identify those individuals at increased risk, can we deliver targeted prevention, and what could such prevention strategies look like? And the detection. So obviously early detection is still something that will massively improve quality of life and survival for patients. As I've mentioned today, I'm going to talk about the prediction aspect. This work was carried out as part of the FORECEE program that has received funding from the European Union and the charity, the Eve Appeal. And our aim was to develop one test that can detect and predict the risk for 4 women's cancers using the epigenome in a surrogate tissue. So the 4 cancers would obviously be the ones that I've mentioned before, so breast, ovarian, endometrial, and cervical. And the choice of surrogate tissue is really important in this context. I've already mentioned before that the epigenome is cell type specific. So the surrogate tissue needs to reflect the tissue at risk in a certain extent. And we also know that hormones play a key role in women's cancer development. So ideally, if we choose a surrogate tissue, it should be hormone sensitive and reflect the tissue at risk. But importantly, it also needs to be easily accessible, because you obviously want to not take repeated biopsies of a sample or of a person for risk monitoring and prediction. And of course, the topic of this talk is about cervical samples. So we've opted to use cervical samples because they are epithelial, or they at least contain a large proportion of epithelial cells, which are also hormone sensitive. And importantly, they are already used in routine screening. So we can take, in theory, residual samples from cervical screening, which includes a large proportion of the women at risk for the other cancers as well, to deliver our WID tests. So the way we went about this was cervical sample collection from healthy women or women with benign conditions, so non-cancerous, and women with cancer, so either breast or ovarian cancer in this specific setting. We've also obviously looked at endometrial and cervical cancers for the overall WID test. And then what we can do is subject these samples to DNA methylation analysis. They're collected in a routine pot, and actually, just out of curiosity, I've included another poll here. This is mainly relevant to people working with cervical cytology, so I don't know if everyone is doing that, but which sample collection devices you use, because that actually has interesting implications for molecular analysis downstream. But yeah, this is really just out of curiosity. So one thing that I think we have done quite uniquely is also look at cell type composition in the samples, but I'm probably going to wait for the result of the survey first. Okay. Cool. So yeah, we actually did our analysis in FinPrep, which seems to be really the majority of what people are using. SurePath also works for molecular testing, but needs some more pre-processing. But yeah, it's interesting to see that you're mainly using FinPrep. So great. Okay, back to the test development. So one thing that really set our approach apart from previous studies was that we accounted for cell type differences. So the good thing about DNA methylation analysis is, as I've already said, we can, it's cell type specific, so you can detect which cells are in your sample based on the patterns. And what that allowed us to do is basically infer the cellular composition of a sample. So these are all cervical samples from either controls or breast cancer cases. And you see the large majority is epithelial cells, as we expect. So these would be likely cervical epithelial cells. But there's also other cells present in the sample, such as neutrophils, monocytes, NK cells, and so on. So it's important to know that because it has implications for the interpretation of the data. What this allowed us to do is look at differential methylation analysis. So basically we checked which CpGs show a difference in methylation between controls and cancer cases, so either ovarian or breast cancer. And this inference of the cellular composition, importantly, allowed us to look at specifically which CpGs showed a differential methylation in epithelial cells or even in immune cells. So this might be a bit abstract at the moment, so I'm trying to demonstrate this a bit further. So here we have a graph of an example CpG. On the x-axis, I'm showing the immune cell proportion for each sample. So for example, this sample here would have roughly 50% immune cells, and the remainder would likely be epithelial cells. But we have a whole spectrum here, really. So some samples are almost exclusively immune cells, which might have, for example, resulted from bleeding. Some have a very high or very low immune cell and very high epithelial cell composition. And then we have the beta value. So that's basically a methylation value for a specific CpG. And that can range from 0 to 1, with 0 being completely unmethylated and 1 being completely methylated in all DNA fragments analyzed. And what you can see that actually a lot of these CpGs have a strong dependence on the immune cell proportion. So in epithelial cells, the methylation is generally lower than in immune cells. But also what we can additionally see is that if you look here, for example, along this line, this is samples with a very high immune cell composition. There's very little difference. Whereas if you look at this end of the spectrum, if you look at epithelial cells only, basically, you see a difference in this beta value for controls in breast cancer cases. So, and this is quite important because actually what it means is if we did not know that there was a difference in the kind of sample composition, we wouldn't have found these differences. And it's also important because if we obviously in a clinical setting take samples, we need them to be, we need to be able to identify breast cancer cases regardless of whether the sample has a high or a low immune cell proportion. So we found differences for both immune and epithelial CpGs. And we were then able to build a model. So essentially what we did, and I'm not going to go in any detail, but we give a machine learning model a certain number of CpGs that we identified as different between controls in breast cancer cases, and then check if it can, given the same CpGs for another completely different set, identify who has breast cancer and who doesn't. So that was the overall model development. We did this separately for ovarian and breast cancer, and also the other cancers that we are interested in, which have not yet been published. I'm going to start by talking about the ovarian cancer. So here we're looking at an independent validation set. So that means these samples have not been used for training or anything. So they're completely, the model has never seen these before, basically. And what we see is that actually we're quite good at discriminating between controls in blue and ovarian cancer cases in red. And this is not strongly dependent on the immune cell proportion. So as I've highlighted, it's important that we can detect them even in samples that have high immune cells and not many epithelial cells. On the right, you see a receiver operating characteristic. So for those who are not so familiar with it, it's a common tool used for evaluation of diagnostic tests or predictive tests, for instance. So it's basically if you were to take any value of this OC index and put a threshold, how many individuals would be basically true positives and false negatives. So the sensitivity and specificity would be calculated. So just as a brief example, so if we were to cut, for example, the WID-OC index at -1 and say anything below is negative and anything above is positive, then we would obviously correctly identify all these controls as controls. So we would have a very high specificity, but we would not, identify very many, so all of these controls that are above the threshold would be falsely identified as positive, so theoretically ovarian cancers. So that would mean it would have a high specificity but a low sensitivity. Vice versa, if we have the cutoff over here, we would have a higher sensitivity, so we would detect a higher proportion of ovarian cancers, but the specificity would be very bad because we would include all of these ovarian cancers in our kind of false positives. So so this is basically across the entire spectrum. We can draw this diagram and we have a pretty good discrimination. So for example, if we look here, so that would be 1 minus 25 specificity. So 75% specificity, we have a 75% sensitivity. And this is outperforming most current diagnostic tests for ovarian cancer, which is quite, given that there's few sort of screening tests that are available at the moment. What we found then quite interesting is obviously we know that the ovaries are in relative close proximity to the cervix. So we were wondering whether the signal that we see is derived from tumor DNA that would be picked up in the cervical sample. Similar to inference of the immune cells, we can also check how much of a tumor DNA we would have in our samples. So we can basically look at high-grade serous ovarian cancer DNA and then see, okay, in our control samples, how much of that would theoretically be in there. We then, when we're looking at controls in ovarian cancers, we were somewhat relieved because it didn't look like there was a very high proportion of tumor DNA. There were 2 samples that had relatively high tumor DNA, but as I said, this was a minority. And we further confirmed this kind of finding when we looked at copy number variations. So in tumor tissue, it's known that there's a lot of genomic instability and rearrangement. So when we look at a high-grade serous ovarian cancer sample, and we look at these are basically genomic bins and copy number alterations. So if it was all 0, there would be no alterations from the baseline genome. And if there's an increase or a decrease, these are shown in red and blue. You see there's a lot of genomic variability and copy number alterations in an ovarian cancer sample. When we look at a cervical sample with high tumor DNA or high inferred tumor DNA based on our analysis, we see again that there is actually quite a lot of rearrangements which might be driven by this tumor DNA. When we look at a sample where we infer that there is not much tumor DNA, the genome is flat. So this was kind of a nice confirmation that, okay, we have some samples that do have tumor DNA, but the majority does not. We were next interested in seeing whether the OC test could also identify other cancers, because that could be indicative of a more kind of epigenetic risk being indicated rather than detecting tumor material. So what we did is we assessed the OC test in women with endometrial cancer versus controls. And this was really striking because actually the discrimination was really, really high, even higher than for the ovarian cancer index. For the ovarian cancer. Likewise, although a less pronounced effect, we also found that the OC index could discriminate controls from women with breast cancer based on their cervical samples. The AUC was lower, so you have less of a strong effect, but it still was able to discriminate. And lastly, we also looked at healthy women with a BRCA1 mutation. So these do not have cancer, but they have a very high risk for breast or ovarian cancer. And even for those, we had a certain amount of discrimination between controls and mutation carriers. So this again indicated that the OC index was indicative of some sort of epigenetic field defect. We were a bit struck by the finding of the high discrimination between endometrial cancers and controls using the OC index. And when we did the same tumor DNA inference for endometrial cancers, which are even closer anatomically than the ovaries to the cervix, we found that actually, yeah, quite a few of these cancers had tumor DNA present, which makes sense because of the anatomical proximity and that the smear test might pick these up. However, what we then did is basically on the x-axis, we look at the tumor DNA fraction for each sample and the OC index. So yes, there was a dependence of the OC index on the tumor DNA fraction with a tendency for a slight increase. So with increasing tumor DNA in your sample, the index would be higher. However, even when we had samples at 0 tumor DNA, there was still a big difference between cancers and controls. So again, this indicated that the OC index is basically kind of a risk indicating signature that we're picking up in the cervical samples. So to summarize the first part of the WID tests, yes, we can detect ovarian, but also even other gynecological cancers using this test. We think it's likely that the test is picking up an epigenetic field defect, identifying cancer risk, especially because there was very little dependence on the actual tumor material. Okay, so that was the first part of the WID tests. I'm now going to briefly also talk about the breast cancer index. So we did the model development essentially analogous to the ovarian cancer and found again in the validation set that the WID-BC index of breast cancer discriminates well between controls and breast cancer cases, and the AUC was actually even higher, so it's 0.81 compared to the ovarian cancer. Again, we were interested in seeing whether the features that we're picking up are driven by kind of tumor features or tumor DNA, or we're picking up this risk marker. So obviously the breast is quite far away from the cervix, so it's quite unlikely that the tumor DNA is present in the sample, but we can't completely exclude it. And what we did to go around this is we looked at publicly available data from the Cancer Genome Atlas and essentially did the same thing for the model development. So we tried to find features in breast tissue that are associated with breast cancer. And when we apply this index that's been developed on actual tumor in the breast, we see that it discriminates really well in breast tissue between controls and breast cancer, but it doesn't at all in cervical samples. So these breast cancer features that are specific to the breast are not really present in the cervical sample. So especially when we compare that to the BC index, then we see there's a big difference. So that was reassuring, and it meant that we were able to pick up current cancers. So far, what I've mentioned a little bit, but not actually shown, is that we have a bit of a predictive performance as well. So with the ovarian cancer index, we've obviously already shown that we have this epigenetic field defect, which is indicative of risk prediction, but we haven't yet looked at samples before diagnosis. So in the for the BC index, we were lucky or fortunate enough to collaborate with partners at the Karolinska Institute, where they biobank essentially all cervical samples that are collected in the Stockholm region, and they are also linked to cancer registries. So we were able to access a subset of samples that were collected in years preceding diagnosis. So the women were all healthy or cancer-free at the time of diagnosis, or at least there was no known cancer. So unfortunately, one caveat to this set was that the storage was not optimal for DNA methylation analysis, which resulted in a lower signal-to-noise ratio. So just looking here in the discovery set, so that was the set I've described before, where we purposely collected samples from controls and cancer cases. We have quite a good quality, but this was significantly lower in the biobank set. Nonetheless, when we looked at the predictive potential of the WID-BC test, we found that there was some signal retained. However, this was really strongly dependent on the signal-to-noise ratio. So at low signal-to-noise ratio, the AUC was quite low, and then at higher signal-to-noise ratio, we had an increased AUC. So we had some discrimination between cancers or future cancers and controls. The confidence intervals are quite wide, so that is a limitation, but we hope to validate this further in the future. And lastly, what we wanted to look at in a bit more detail is actually some functional work, because a lot of what we've done so far was really rather descriptive. But we were particularly interested in progesterone. So progesterone is known to be involved in breast cancer development. And we thought by investigating methylation at so-called progesterone receptor binding sites, this could be indicative of higher lifetime exposure. So meaning basically if you have a DNA strand which progesterone can bind, and you have a lot of progesterone, this site would be occupied more and thus prevented from being methylated. And then basically this lower methylation than expected would indicate a higher risk for cancer. So to look at this in a bit more detail, what we did was to compare methylation in normal tissue, normal breast tissue compared to breast tissue that was adjacent to a cancer. So the tissue itself was normal, but it was next to a cancer at these progesterone receptor binding sites. And we did the same in breast cancer cases and controls for cervical samples. And basically what this was aimed to do is look at if there's any similarities in these two tissues in terms of the progesterone receptor binding sites. So when you look at, for example, x-axis 0, y-axis 0, this would indicate that there is neither a differential methylation in cervical samples between breast cancer cases and controls, nor in breast tissue from adjacent to normal. However, some of them obviously change direction in one, but not the other. But what we do see is this very strong skew towards a loss of methylation in both the breast tissue and the cervical samples, which was a lot higher than would have been expected by chance. So these are a set of CPGs that lose methylation in both breast tissue that's adjacent to a cancer and cancer cases and controls. What we wanted to do then was to assess in a different set whether this was again correlated with increased risk. And we basically looked at the mean methylation of this group of hypomethylated progesterone receptor binding sites in healthy controlled breast tissue. So these would be healthy women that have undergone, for example, plastic surgeries and had breast tissue removed, or risk-reducing surgery tissue from BRCA mutation carriers. And you see, even in those, we see a loss of methylation, which again might be associated with risk. So this has led us to the kind of overall thinking or hypothesis that basically the progesterone receptor binding can be indicative of breast cancer risk. So if you have a hypomethylation, this would mean that there's a lot of binding of this receptor, which could indicate higher breast cancer risk, and we can then measure this. So in the breast, it would drive cancer formation, whereas in the cervix, it might not do so, but we can still measure this effect because the cervix is hormone sensitive, and it might therefore be a surrogate marker for cancer risk. Yeah, so basically we see similar epigenetic patterns in breast tissue at risk and cervical samples, which was very reassuring because this was exactly what we aimed to develop in the first place. So in summary, we have developed the WID tests, which I now realize I've not explained the acronym. So it stands for Women's Cancer Risk Identification. And they are based on cervical samples, which can be done in, for example, routine thin prep storage pots. And these can then undergo DNA methylation analysis. The same sample can be used for any of these tests because they're based on the same kind of data format. We can detect breast cancer and ovarian cancer, and very likely also predict the risk for either of them. Now, some outlook and future research questions. Something that I'm particularly interested in is the dynamic monitoring of these indices in preventative measures. So can DNA methylation be used to quantify, so to say, the effectiveness of risk-reducing measures? We are currently doing a clinical study where we follow individuals who either stop smoking or undergo intermittent fasting and see how this changes their WID indices. And this will also be carried out in a more large-scale manner in the future. We also have in planning, obviously, the large-scale validation of these WID tests in prospective studies, as we are hoping to roll these out in the clinic and offer these to women in Europe very shortly. And of course, we are also interested in the evaluation of self-collected samples, mainly because this might enable higher take-up for screening purposes. But also, I think the last 2 years have shown that sometimes cervical screening has to be halted for reasons that is out of everyone's control. But we wouldn't want to give up screening. So this is something that we are quite interested in evaluating. And just as a final poll before I stop, I just wanted to ask if anyone has had experience with self-collected cervical samples, if you work in the cytology field. I think this is something that is at the moment done in Sweden. So our collaborators at Karolinska are working with those, but I don't know how common it is in the UK yet. Okay, 20%. That's interesting. Yeah, so it would be interesting to talk about any experiences if there's some time in the Q&A session. Okay, great. So I'm basically at the end of my talk. All of this work has been led by Professor Martin Midschwenter, who's a professor at the University of Innsbruck, UCL, and also Karolinska. And it's really his vision and ambition that has been driving these massive projects. And as you can see, we are a big team, so none of this work could have been done by one group. We're very lucky to work with really inspiring people all across Europe. Last but not least, I also want to thank our funders. So a big proportion of this work has been funded by the European Research Council and the European Commission. We have a very good relationship with the EVA Peel, who are really extremely helpful. And we also have received funding from the Land Tyrol in Austria and the University of Innsbruck. And this is a small part of our team in that picture shown. So yeah, thank you. And I'd now like to welcome any questions.
Oops. Thanks, Ciara. That's really interesting. I really enjoyed that talk. Thank you. There are a few questions in there. I'll just go in order. Can the WID test be used for other cancers such as lung, pancreas, or colon?
Yeah, that's a very good question. So it's actually something that we're really interested in looking at. At the moment, we're a bit limited by the fact that we don't have the sample collection for these types of cancers. In theory, we do believe it's possible because especially colon and also pancreas are also epithelial-derived cancers. So we do believe that is possible. And yeah, it's something that we're basically working on almost.
That's grand. Another question there is, can the WID test be used on blood samples?
Super question. Yeah, thank you very much for that. So we have had some matched blood samples from individuals where we also did the cervical samples. I don't have the data in the presentation, I think, but the discrimination was a lot less. So even the same test. Okay. Now this could be because obviously the test was developed on cervical samples. Nonetheless, what we have seen throughout, and this is also why we initially decided to use the cervical samples, is that the variability in blood methylation is a lot less just because of the sample composition. So because it's essentially only immune cells, the signal is just a lot weaker. But yeah, I mean, this is something that people are doing. So. But yeah, for us, it was the type of sample, the cervicals were the strongest signal, so to say.
Okay, then this is a question then, as in understanding, I'm sorry. Yeah, are the results independent of specific histological type of breast and ovarian cancer?
Yeah,
yeah, and the other part of that is, do they detect borderline or low-grade serous ovarian tumors?
Yep, so that's a very good question. We have aimed to include mainly poor prognostic cancers, both for ovarian and breast cancer. So we don't want to overdiagnose, but obviously we want to pick up all relevant cancers. I think we are, from what I recall at the top of my head, we detected all poor prognostic cancers, so that the high-grade cancers. We were also, so if it was an aggressive cancer, we would have detected it. For borderline, the detection was slightly lower. Okay, great. Actually, this was one I was going down the road of. What would be the proposed management for patients identified as a risk or at risk? Yep. So this is obviously a big question that we are working on at the moment. So this is part of the large-scale study that we want to do, because obviously, you know, it's a bit scary as a patient if you receive, okay, you have an increased risk, what do you do? So one aspect would be that we want to invite these patients to more frequent screening, or for example, invite them to MRI, some sort of scanning, but it might be combined with other molecular tests that we are developing at the moment that can detect early cancer formation. The other option, especially if we do not detect a cancer, would be to offer preventive measures, some of which we are currently, so a paper that will appear quite shortly, hopefully. So yeah, but it is something that needs to be really well defined before anything can be used in the clinic. Okay, and then a question here about immune cells in the cervical sample, that these are non-epithelial cells. And would there be any value in trying to remove them for analysis? Yeah, so again, a really great question. Thank you, guys. We have, there are options to remove epithelial cells or debris. So I think actually SurePath is going along this route a little bit by trying to enrich for epithelial cells at least. But actually something that we have seen is, despite our original hypothesis that the epithelial cells are only what we want, we actually have a signal. So there's a large part of the signal as well coming from these immune cells in the cervix. And this is also interesting because we find that there are differences between, for example, immune cells in the cervix and in blood. So even though in blood the signal is not very, very high, in these immune cells in the cervix it is. And I think this is quite interesting. So this is something that people are starting to appreciate more and more.
Okay, just as an aside then, could that be something to do with the field change in relation to those immune cells at that site?
Exactly. So, I mean, at the moment, we don't know. It is very likely something to do with the interaction between the cell types in the tissue. So this is known for other tissues. So, for example, in the liver, it's known that the immune cells that migrate there during development, they basically stay there and they're their own self-renewing stem cell pool, whereas the blood immune cells, they don't really interact normally. So it is super interesting, and yeah.
Okay, and then just a question regarding cell sampling. That working with cell sampling, obviously this is a swab. Would that work as well due to reduction in epithelial cells?
Yeah, so there is some initial work we have done, not for these specific WID tests, but based on PCR. And because we do also have hormone sensitive epithelium it is from the vagina rather than the cervix itself. But it does seem to still retain the signal. But yes, it is something that needs to be carefully monitored because obviously the sample composition is slightly different.
Okay, I have a question as well, just in relation to the HPV status of the women with these cervical samples. Does that have any influence, particularly with the epigenetic aspect and methylation markers associated with those?
This is something we are still looking at. So in terms of the tests that I've shown today, we do not see any dependence on the HPV status as far as I'm aware. But of course, viral infections can change the epigenome. And we actually have some interesting upcoming work on this and how kind of the virus might render the cells more prone to progression for cancers.
Okay. We're getting some really good positive feedback here saying thank you and well done. I have one final question then again, and it comes back to the breast work that you do. You're looking at progesterone hormone effect. I'm just thinking of progesterone only contraception pills and other modes. Does that have any influence as well, or could that kind of?
It is tricky. So it does seem to be the lifetime exposure. So obviously this might depend on contraceptives. It's not clear if it's only based on that or if it's also some intrinsic factors. So in some upcoming work, we have actually shown that, for example, BRCA gene mutation carriers have a higher kind of overall progesterone, higher overall progesterone levels throughout the menstrual cycle than controls. So this is something that, we're not entirely sure how this works. At the same time, for example, I think oral contraceptives are a good example of kind of a dual effect because they might have a slightly increased breast cancer risk, but at the same time, they're associated with a strong effect in ovarian cancer reduction. So it's really difficult to kind of disentangle all these effects. But it does seem to be also individual factors that contribute to that.
Okay, that's great. And yeah, one of the last comments there, a great talk. Hope the studies proved to be developed into a great screening tool.
Thank you. Yeah.
So thank you. I'd like to take this opportunity to thank Dr. Herzog for presenting on her work, and we look forward to your upcoming publications on the studies as well.
Yeah.
So thanks everyone. The presentation has been recorded.